The brain is made out of many different kinds of cells called neurons. These cells come in literally hundreds to thousands, we don't know for sure, different sizes, shapes, and connectivities. So if you're trying to solve the brain as a circuit, you need to be able to see how information is flowing. Scientists have used optogenetics to activate different sets of cells in the brain and found cells that can trigger behaviors ranging from violence to emotion to memory recall and everything in between. So for the first time, we can really tease apart how specific kinds of cell, even those deep, deep in the brain, can initiate something as complex as a behavior. Karl Deisseroth and I collaborated to develop the use of microbial opsins for the control of neurons using light. These are molecules that grow in organisms all over the world and convert light into electrical signals. So Karl and I started asking around to find molecules that, if we put them into neurons, might allow us to control them using light. Back in the summer of 2004, we got lucky. Karl put one of the genes that we'd asked for into some cells, neurons, in a dish. I shined light on the neurons, and serendipitously, the neurons could fire electrical pulses with the same temporal precision as the brain itself. So this was quite lucky that nature provided a molecule which was perfectly matched to the problem of understanding how brain cells influence behavior. Initially, people were a bit skeptical about optogenetics. I remember looking for a faculty job and finding that some places didn't really think that neurotechnology was ready to become a real discipline, it seems. There was, of course, a period of many years where people started to learn how to apply it to different systems, different structures within the brain, different parts of the nervous system. And over the first 3 or 4 years, the momentum started to gather. And then it started to accelerate, and to the point where we have now sent these molecules out to literally thousands of research groups in academia and industry. People are now using these tools to study all sorts of different behaviors and even diseases. But in the beginning, I think maybe people thought it was too good to be true. Here's a molecule from the natural world, and you just put it into a cell and control it with light. But it worked. Optogenetics lets us control neural circuits very precisely. Next, though, we need to be able to see them, both their structure, how information flows through the wires of the brain, and also the dynamics, to watch the high-speed electrical pulses in the brain that help it compute. We're now working on all sorts of different technologies to help us map the fine wiring of the brain, and also to see the computations in action. The brain is very dense. If you took a human brain and somehow expanded it until it was the size of a city block, the individual connections between neurons would be the size of a grain of sand. So we need ways of mapping enormous circuits in the brain while preserving the nanoscale fidelity of those connections.